So welcome again guys, welcome again in another video of our recombinant DNA technology video lecture series. So today as promised we are going to talk about frequency of recognition site. Okay, so let's discuss about this in a very normal term. I hope you all know about Lionel Messi, who he is, right? So suppose he is going to take a free kick, right? So what is the probability that he will make it or he will convert it into a goal, right? So or you can say suppose there is a super over is going on in cricket, and what is the probability that Dhoni will hit on the how many sixes that Dhoni can hit in six balls, right? So frequency of recognition site is something like that. We all know that restriction endonuclease will cut a double helical DNA fragment, right? So restriction endonuclease have the ability to cut double helical DNA fragment of a specific sequence that they will recognize, right? And this specific sequence we termed as what, recognition site, or we can say recognition sequence. Now let's talk about, now suppose you have a very large DNA fragment, right? And you need to find out what is the how many restriction site will be there for a particular restriction endonuclease. Let me clear it more. Suppose this is a long chromosome. Okay, now suppose this is your restriction endonuclease. Okay, so it will recognize a particular restriction site here, and it will make a cut it, and there can be multiple restriction site in overall chromosome. Okay, so in the frequency of recognition site, that simply wants to tell us that how many recognition site are there in the overall the chromosomes. Okay, that our restriction endonuclease have the ability to recognize. Okay, so I hope now it is clear to you. So the number of recognition site for a particular restriction endonuclease, the number of recognition site, mark it, for a particular restriction endonuclease in a DNA molecule of known wavelength can be calculated mathematically. That what I have told, the number of how many recognition site will be present in a particular chromosomes that will be recognized and cut by restriction endonuclease. Okay, so let's come here. So first of all, let's clear one thing, that the restriction endonuclease restriction. Okay, restriction endonuclease have the ability to recognize either four base pair sequences or six base pair sequences. Okay, so in both the cases there can be two possibilities. In one possibilities there will be same composition, and in another case different composition. In case of same composition we have ATGC of all of 25%, right? Now but suppose this is a restriction endonuclease HAE3. Let's us revise that how we can nomenclature a restriction endonuclease. So HE3, in that restriction endonuclease, H is the genus, A and E is our species, and 3 is the order of enzyme that is isolated, or you can say purified from that particular bacterium. Okay, and this is our restriction site that this HAE3 gonna recognize. Okay, so and this simply tell us the position in which HAE3 will make the cut. So as if you watch my previous videos, so you can easily interpret that this is a blunt cut, and this will ultimately produce the fragments that have blunted. Okay, so we revise our previous lectures. Now they are telling us that this HEA3 have restriction site in a DNA molecule which have a overall length of approximately 1 lakh base pair. Okay, so we need to find out among this 1 lakh base pair, how many restriction sites are present? Okay, so suppose this is the overall this is the overall DNA molecule of 1 lakh base pair, and here we have the probability of occurring G will be 1 by 4. Why? Because we have 4 nucleotide, and in one place, according to the rule of probability, as all are in equal amount, we can say the probability that G will be occur is 1 by 4. Okay, and again G will be occurring, as they are telling us, will be again 1 by 4. In case of C, that is again 1 by 4. In case of C, that is again 1 by 4. Okay, so we can ultimately conclude by multiplying all four that 1 by 256 that will be our answer. So what this value telling us? This value simply telling us that there is a 1 recognition site in every 256 bases. What it is? Suppose this was our 1 lakh base pair chromosome. So here every 256 base pair, suppose this length is 256 base pair. Okay, so there can be a 1 restriction site. That is what these math problems want to tell us. Now if they give you the overall and this is also average fragment size. Okay, and if they give you the overall genomic length, then you need to use unitary method to find out how many restriction sites are present. Okay, that means in 2056 base pair there is a 1 base pair recognition site, or you can say there is a 1 restriction site. So for 1 lakh base pair, how many restriction site will be there? That is 390 base recognition site. So I hope this is clear to you. In similar way, in case of 6 base pair sequence, you can apply the same funda to a seat. Now in case of different composition, we are now going to talk about. So for doing such kind of problems, when they are telling you that GC component is 60% and 80 is 40, what we will need to do? For doing such kind of problems, you need to know about Chargaff's rules. That is simply purine is equal to pyrimidine. Okay, that means A plus G will be your T plus C, or you can say A is equal to T or G is equal to C. These are the two rules. Amount of purine is exactly equal to amount of pyrimidine, or you can say A is equal to T and G is equal to C. You need to know these two rules when to process solve such kind of problem. Suppose you have a question that HIN3 is our restriction endonuclease that have the ability to cut the record restriction site. Okay, so they are telling us that in that, again let me take my headphone. I will make it as a chromosome. So this is your chromosome, and HIN3 is the restriction endonuclease. It will recognize a site here. Okay, so they are telling us about what how many restriction site will be there, and they also give us that in that particular chromosome there is GC content is 60%. That means amount of GC is 60. And in case of same composition, we are considering that GC component is 50%. We will do a math problem and it will be more clear to you. So let's move to the math question first. So welcome again. So let's read the question first. Okay, the restriction endonuclease HIN3, that is recognizes the sequence AAGCTT. So what they are telling us, this is a 6 base pair sequence, right? 1 2 3 4 5 6. If the genomic DNA of a random sequence is cleared with HIN3, so what will be average site of the fragment produced? They are telling us about average site. Okay, that means we need to talk about in what the base pair we have 1 restriction site. Okay, that means we need not to divide it with the overall genomic length, and they do not give us the overall genomic length. Okay, so they lead simply wants to know something like that. So we have to tell that in certain base pair there is a 1 base pair recognition site, or you can say there will be occurrence of one restriction site. Okay, so they also provided us that if the GC content of genomic DNA is 50% or if the GC content of genomic DNA is 40%, that means they are simply telling us about different composition. Okay, so in case of GC component, the probability will be as they're telling us about 6 base pair, and in case of GC component 50%, that means A will be 1 by 4, G will be 1 by 4, T will be 1 by 4, C will be 1 by 4. So we can simply conclude it that the answer will be 1 by 4 to the power 6. That means every 4096 base pair there will be a one recognition site. Okay, that what I have written. Therefore, in a DNA of random sequence, a Henry site might be expected an average every 4096 base pair along the molecule. Okay, so that is what they want to tell us. Now again, if the GC content of genomic DNA is 40%, okay, that means AT content is 60%. That means composition A 30, T will be 30, G will be 20, GC will be 20. Okay, I hope it is clear to you. Then for restriction side, the probability of occurrence will be, we will again multiply them and we will have something like that. That means there will be a one restriction site for every 3086 base pair along the molecule. Okay, that means for A 3 by 10, A 3 by 10 again, G 1 by 5, C 1 by 5, 3 by 10, 3 by 10. So we will have our answer. So I hope this video lecture series is very clear to you, and if you have any problem, okay, regarding this topic, you may ask me in our Facebook group, and you can also ask me in the comment section. Okay, so I hope it is clear to you, and in our next video we will do some MCQ problems from CSI and NET regarding this topic. Okay, and then we will move to our various type of restriction endonuclease, where we are going to talk about type 1, type 2, type 3 restriction endonuclease, and that will be going to be the last video for restriction endonuclease. Okay, then we will move to the N modification enzymes. So I hope you will enjoying our recombinant DNA technology video lecture series. Thank you.